Hello, today I'm going to talk to you about protein synthesis. Syn means to put together, so we're going to look at how proteins are made. Remember that proteins are the workhorses of cells, so they do practically everything for cells, and they are made of 21 different amino acids. The order and the combination that these amino acids are strung together will determine the shape of a protein, which will then determine its function in the body. The order of these amino acids is going to be coded for by nucleic acids like DNA and RNA, and we're going to talk about how that works today. Proteins do all of the work for the cell, and therefore they have a lot of different functions. These are just a few. The number one function I want you to remember for proteins is that they act as enzymes, which catalyze reactions. And this means that they're going to speed up reactions by lowering their activation energy so that the reaction can happen at body temperature instead of some outrageously high temperature that it might need outside of the body. Enzymes fit with their substrate or reactant like a lock and key. And so each enzyme can only do one type of chemical reaction. Now, your body needs to do all kinds of chemical reactions. And since you need one enzyme for every part of that reaction, you can make over 20,000 of these enzymes. Proteins can also serve as chemical messages between cells, and we call these hormones. Now, a hormone is basically just a molecule that one cell sends to another to influence that cell's behavior to make it do something. Proteins can also provide structure for bodies. For example, the hair and the nails in your body are made of keratin, which is a type of protein. Your cytoskeleton in your cells is made of proteins. The muscle fibers in your body that help you move are made of proteins. All kinds of structural components. Proteins can serve as antibodies. Anti means against, and these are little molecules that are going to help fight against infection. Now, they do not actually kill the pathogen themselves, but they can mark the pathogen, the bacteria, or the virus, to be destroyed by your cells. They can also transport materials. We talked about how proteins are embedded in the membranes of cells, and they can actively transport molecules against their concentration gradients. They can also open up holes in that plasma membrane so that large and charged molecules can get through by diffusion. The order of amino acids is going to be coded for in the DNA and the RNA, your nucleic acids. And these nucleic acids are made of nucleotides, which we can see here. Nucleotides have three parts: a sugar, a phosphate, and one of these five bases. And the sugar and the bases are going to be different depending on the type of nucleic acid that we have. We can see DNA here. It is a twisted ladder shape. We call that a double helix. The backbone of this DNA is going to be made of alternating phosphates and sugars. And since this is DNA, we're going to be using deoxyribose sugar. That's what the D stands for, deoxyribo. We can see that our bases are turned into the middle of this helix, and those are A, T, C, and G, and they've been color coded for you here. And you can see that A and T always go together, and our C's and our G's will always go together, and that is for DNA. If we look at RNA, it's a little bit different. It's not a double helix, it is just a single helix. It has a backbone that is still made of alternating phosphates and sugars, but this time we're going to use ribose sugar instead of deoxyribose. And we can see that's what the R in RNA stands for, ribonucleic acid. Our bases are, we still have four bases, but we are using U instead of a T. We still have A, G, and C, and you can see those have been color coded for you. So in RNA, we have no thymine, which is red, so there's no red here. In DNA, we have no uracil, which is orange, so no orange here in this DNA. Now, your base pairing is going to be a little bit different when it comes to RNA, and we'll look at that here in just a few minutes. Since there is no T in RNA, you're going to use a U instead, and we'll talk about how that works here in just a second. So there are three different types of RNA, and all of them are going to be used in protein synthesis. The first type is called mRNA, or messenger RNA. This type of RNA is going to carry information. It's going to carry a message from the nucleus to the ribosome, and the message it's carrying is how to put together this protein. So you can see the mRNA here. It's a single strand of nucleotides, and it's going through this big green ribosome. We also have tRNA, and the T stands for transfer. And this type of RNA is carrying amino acids to the ribosome. It is transferring those amino acids from the cytoplasm to the ribosome. You can see your tRNA here. It's carrying an amino acid. It's going into the ribosome, and these amino acids are being linked together in a chain to form a protein. We also have rRNA, and the R stands for ribosomal, and this type of RNA makes up part of this green ribosome itself. Again, all of these are going to be used in protein synthesis. Protein synthesis is simply the process of making a protein from a gene. A gene is a segment of DNA that codes for a specific type of protein. And remember, you can make over 20,000 different types of proteins in your body. There are two parts to protein synthesis. We have transcription and then we have translation. And these happen in this order. So you can remember it because it's alphabetical. C comes before L. So transcription happens first and translation is going to happen second. The general idea in transcription, DNA is going to be used to make an mRNA message. And in translation, this mRNA message is going to leave the nucleus and go to the ribosome to make a protein. So DNA to RNA, transcription, RNA to protein is called translation. Let's dive a little deeper into this concept of transcription. Remember the overall idea, DNA to mRNA, but this does not mean that we are turning DNA into mRNA. We are just taking the instructions to make the protein that are written in the DNA and rewriting that into an mRNA language. Well, why do we need to do this? DNA is trapped in the nucleus of that cell. It cannot get out, but it needs to send that message to make the protein out of the nucleus as an mRNA molecule. So why not just use mRNA to be the instruction manual in the first place? Well, DNA is a more stable molecule. So over time, it has evolved to be the instruction manual for proteins because it can hold on to that protein code better, okay? So again, this DNA is trapped in the nucleus, so that's where our transcription is going to happen. We have our double-stranded DNA, and in order to make a protein, we have to find the segment of DNA that codes for the protein that we want to make. It's going to be on one side or the other, all right? Could be either one. Now, this DNA is going to be used as a template to make the RNA strand through base pairing. So once the DNA is separated, this enzyme is going to come in and it's going to start making RNA by base pairing, just like we did with DNA replication. Only this time, A's in DNA are going to pair with the U's of an RNA nucleotide. So A will go with U, C will go with G, G still goes with C, and when we see a T in the DNA, it's still going to be paired with an A RNA nucleotide. So the only difference is in place of writing a T, we're going to write a U instead. Now that's a little bit different than replication. We've talked about replication before. So let's compare transcription and translation. With DNA replication, we are copying DNA. We are making two identical DNA molecules that are going to be double-stranded. To do that, we use an enzyme called DNA polymerase. Poly for many, mer for part, ase for enzyme. So DNA polymerase is an enzyme that puts many parts together to make DNA. And this is going to give you two identical double-stranded DNA strands. With transcription, we're going to use a different enzyme, RNA polymerase. We saw it on the last screen. So this time we are using an enzyme that puts many parts together to make RNA. And we're doing this by looking at a DNA strand. Now with DNA, we made a double helix, had two identical copies of DNA that were both double-stranded. With transcription, we're only making one strand of nucleotides, and that's going to be your RNA molecule. Now, another big difference is in place of thymine, with RNA, we are using uracil. So where you would normally write a T with DNA replication, for transcription, you're going to put a U. Transcription is not as complicated as you might be thinking it is. This cell is living in a certain environment with a certain set of proteins. And if that environment changes, then the set of proteins it's using might change as well. So the environment is going to trigger a chemical signal that's going to be sent to the nucleus to kick off protein synthesis. And we're always going to start with transcription. Now the DNA has the code for all the proteins. So we're going to have to find the gene, the segment of the DNA that we need to make that particular type of protein. Once it's found, that portion of the DNA is going to be separated, and we see that happening here. Once that DNA is separated, this RNA polymerase can then get to the gene that it needs to copy into an mRNA language. So RNA polymerase is going to bind at the start of that gene. Then RNA polymerase is going to attach these complementary nucleotides by base pairing to make this RNA. And again, in place of T, we're going to be using a U. This mRNA is going to be produced. And once we have the mRNA that we need to make the protein that we need, this DNA is going to reconnect until it's needed again. Okay? Let's practice transcription. You have a DNA sequence of bases that reads like this. Now this RNA polymerase is going to come through and it's going to add nucleotides based on this sequence, and it's going to use base pairing. Now remember, in place of a T, we're going to be using a U. So what's going to go with A? U. T is still going to go with an A. C will still go with G. C will go with G. T still goes with A. G goes with C. A, we don't have any T's, so we're going to use a U. T still goes with A. And A, again, is going to go with U because we have no T's. Okay, so this is our DNA strand, and we know this because it has these T's in it. This is our RNA strand, and we can tell it's RNA because it has these U's in it. Okay, so very simple little process, and this is called transcription, DNA to mRNA. Once the instructions for making a protein have been converted from DNA into mRNA language, that mRNA is going to leave the nucleus through a pore. Pores are little holes in the nucleus. We can think of them like doorways that this mRNA is going to use to leave. So as soon as it leaves the nucleus, that mRNA is going to be in the cytoplasm. And from here, it's going to find a ribosome, and ribosomes make proteins. There were two places in a eukaryote that we said you could find a ribosome. One was the cytoplasm. The other was the rough ER. So this mRNA is going to find a ribosome in the cytoplasm, and depending on the kind of protein it's trying to make, that ribosome might go to the rough ER to finish making its protein. It just depends on the type of protein. And any ribosome can make any type of protein. So the location of where this protein is going to be made is determined by the code in that mRNA. Now the instructions on this mRNA molecule are going to be read by the ribosome in groups of 3 bases that we call codons. And that's why I've always written my bases in groups of 3. I want us to get used to this idea of grouping them together by 3s because these are called codons. Each codon is going to code for a certain type of amino acid. And so we have a codon chart. Now this codon chart is universal, which means it does not matter if we're trying to make a protein in a bacteria, a plant, us, yeast, it doesn't matter. It's going to use the same kind of code. So we can see that there are 21 different types of amino acids, but there are 64 different codons. There's 64 possible groupings of 3 bases that we can have. So there are some amino acids that are going to be coded for by more than one codon. Now, there are also some other things I want you to notice about this chart. There is something called a start codon, and this means that these 3 bases, AUG, are going to start the production of every protein. There's also 3 codons that code for stop. So when the ribosome runs into one of these 3 groupings of codons, it's going to stop making the protein. There is a tRNA, but it does not carry an amino acid, and so it's going to end that protein chain and stop the synthesis of that protein. Also notice that these amino acids are abbreviated with the first 3 letters of their names. So we don't have to worry about calling this proline. You can just say it's PRO. You don't have to call this serine. You can just say it's SER. And that tells you what kind of amino acid you're dealing with. So an overview of translation, we're going from mRNA to an amino acid sequence. And this is going to happen in the cytoplasm within the ribosome. So just a general idea of how this works, mRNA is going to leave the nucleus and go to the cytoplasm. That mRNA is going to find a ribosome, and that ribosome is going to read the mRNA bases in groups of 3 that we call codons. Each codon is going to fit with a certain tRNA that carries a certain amino acid. So this little group of 3 that we see here on the tRNA is going to match by base pairing to this codon. And we call this an anticodon. And I remember this because anti has T in it. So the anticodon goes to the tRNA. And this anticodon is going to determine which type of amino acid it's carrying. Now keep in mind, we read the mRNA sequence when we use a codon chart, but the anticodon is how this is going to determine the amino acid sequence by base pairing. tRNAs, or transfer RNAs, are going to carry amino acids to that mRNA sequence in the ribosome. Now a tRNA looks like this, kind of like a T or a clover leaf. This is their 3-dimensional shape, okay? And this tRNA has these 3 bases here that we call anticodons. Anti has a T in it, T for tRNA. These anticodons are going to match to the mRNA codon. So if I were thinking about the mRNA codon this would match to, it would be CUU, because this anticodon is going to match up by complementary base pairing. Same way we've been talking about matching up bases this whole time. So the sequence of the anticodon is going to help determine the shape of this tRNA, and that's going to determine the amino acid that it carries. Remember that codons are found on the mRNA. We're not looking at the tRNA when we're reading this chart. This is a codon chart, not an anticodon chart. And reading this is pretty simple. We're going to find the first base in this row here. So everything in this row is going to start with a U as the first base in the codon. Everything in this row is going to start with a C as the first base in that codon. This row, every codon is going to start with an A, and in this row, everything's going to start with a G. Okay. The second base in the codon is found up here at the top. So the second base in the codons in this column are all U's. The second base in these codons in this column are all C's. If we look straight down. These are all A's as the second base in these codons, and these are all G's. So we'll practice with a few codons here. You have AUG to start with. So we're going to find A, and we're going to find U, and where those crosses here. So we find AUG. Means we're going to start with methionine, MET. GUC, find our G and our U. So we're right here in this box. GUC codes for valine, VAL. ACC, we have A and C. So we find the cross. It's going to be right in here. THR, we have threonine. We have GAC, we have G, A, they cross here. GAC, aspartic acid. UCG, here's our U and our C. From there we find UCG, and we have SER, or serine. UUA, we have U and U, so we're here. And UUA is going to code for leucine, LEU. Then we have UAA, find our U, our A, they cross here. UAA is going to code for stop. So you'll have some more practice with this as we go. We've talked a lot about ribosomes, which make proteins. So the ribosomes do this by assembling the amino acids, putting them together in the correct order to get this protein to fold into the right shape. So there are 2 subunits for a ribosome, and those are going to come together around an mRNA when we need to translate that mRNA. So these subunits are made of rRNA, or ribosomal RNA. And this rRNA makes up these subunits. It's going to make the structure of this ribosome, and it's going to help it with its translation. Now, there are differences in the ribosomal RNA between different species, and there's some very distinct differences between prokaryotes and eukaryotes when it comes to the ribosome. So we can get an idea of the evolutionary history of organisms by looking at their rRNA sequence. As this ribosome is chugging along this mRNA, and it is the ribosome that does the moving, it's bringing in certain tRNAs carrying specific amino acids based on this codon and anticodon matching up by base pairing. Now, there's enough room in a ribosome to fit 2 tRNA molecules at one time, but there are 3 sites that these tRNAs are going to pass through. And you can remember these if you want, because the first letter in each spells APE, A-P-E. But I don't care if you know their names, just kind of know what's going on here. So the tRNA is going to enter in the A site, and as those tRNAs are shifted from the A to the P site, there's going to be a peptide bond made. And this is the important site, the peptidal site. Remember, a peptide bond links 2 amino acids by joining the carbon in one amino acid to the nitrogen in another amino acid. And that's going to grow the chain. Once this peptide bond is made, the tRNA is free of this amino acid, and it's going to leave immediately through the E site, or the exit site. Once this empty tRNA is released from the ribosome, its amino acid has been used. It's going to go out to the cytoplasm, and it can then find the same type of amino acid that it just dropped off, and it can be reused again as needed. So the ribosome is going to continue to attach these amino acids on these tRNAs until it finds a stop codon. Now, there'll be a tRNA for that stop codon, but it's going to be empty. And so there will not be an amino acid to form that peptidal bond with, and so the chain will be broken, and it will leave that ribosome to then fold into the shape that it needs to be. So this whole idea of making a protein can be thought of as gene expression. Gene expression, we are simply taking this gene, this small piece of DNA that codes for a protein, and we are using it to actually make that protein. Gene expression is a little different than regulation. Gene expression is just taking that information and using it to make the protein. Gene regulation goes a little further. Gene regulation talks about when to make the protein, the amount of protein to make, in addition to the type of protein. So this is a little bit more to regulation. And this idea of gene expression brings us back to our central dogma of molecular biology. Dogma is a central idea, a belief system that seems to work pretty well when it comes to molecular biology. We start with this DNA molecule. We convert that information into mRNA during transcription. This is done by RNA polymerase. If we are taking that mRNA molecule and converting that information into an amino acid sequence to make a protein, this is translation, and this is done in the ribosome. Now, we can also take this DNA and replicate it using this DNA polymerase, and that's just copying DNA. So if you know this central dogma, this DNA to RNA is transcription, RNA to protein is translation, you're going to do just fine with this segment. So the idea of gene expression brings us to differentiation. Differentiation has the word different in it, and there's a reason for that. This only happens in multicellular organisms that have a lot of different kinds of cells. But all of the cells in a multicellular organism have basically the same DNA. So every cell in your body has the same instruction manual for making proteins. So why do your eye cells look different than your heart cells and do different things? Well, they're not going to be identical in how they express these genes. Only a fraction of the genes in that DNA segment are going to be expressed in a certain type of cell. For example, your eyes need to detect light. So they're going to make the protein to detect light. Your heart cells, they don't need to detect light. So they're not going to make that protein. They need to pump blood. So they're going to make that set of proteins. Your eyes don't need to pump blood. So they're not going to make that kind of protein. They're going to make different things. So only a fraction of genes are expressed in a certain cell at a given time. And this also goes back to gene regulation. This is a very complex mechanism in cells that determines whether or not a gene is going to be expressed. And this has a lot to do with the environment that that cell is in at a certain time. The process of gene regulation is pretty complicated, but we're going to simplify it here as best we can. The proteins produced in a cell depend on the environment that that cell is placed in. And a lot of that has to do with where this cell is in your body. And I got a couple of examples for you here. Antibodies are proteins produced by cells when you detect a pathogen in your body, a virus or a bacteria, something that's going to make you sick. Now, the more this pathogen is detected, the more antibodies you're going to make. So if you're exposed to a disease causer frequently, then you're going to be able to fight it off better because you're always going to keep this set of antibodies for that pathogen around. But if you're not exposed to this illness frequently, then you're going to stop producing those antibodies. So when you do come in contact with it, you're going to have to start over and make these antibodies again. And that's going to take more time for you to get better. Here's another example. In an embryo, as it's growing into a mature organism, if these cells are growing near cartilage, they're going to become bone cells because they're going to pick up on this tough matrix that they're growing around. And that's going to trigger them to become this hard material we call bone. Now, gene regulation is controlled by the amount of time this mRNA spends in the cytoplasm. Multiple ribosomes can attach to this mRNA at the same time. So as soon as this mRNA leaves the nucleus, it's being degraded. It's being eaten away by different proteins. Now, we have a couple of tricks to make sure that this mRNA stays around for the right amount of time so that we can make the right amount of protein. The first trick is we add this cap, and this cap is going to keep this mRNA from being eaten away from this direction. So it can only be eaten away from over here. So this mRNA is going to have a poly-A tail added. That just means it's a long series of A bases. And the longer this poly-A tail, then the longer it's going to take to eat through and get to this coding region where the protein is actually being coded for. So we're being eaten away from this end until we get to this coding region. Once this is eaten away, then the protein can't be made anymore. And that's going to determine the amount of protein that can be made. So what happens when this DNA coding for proteins becomes damaged? Well, we can wind up with cancer. And cancer affects 1 out of every 5 people in this country. The only disease that kills more people than cancer is heart disease. So cancer is the second leading killer of people in America. Now, cancer happens when the proteins that control the cell cycle, how quickly a cell reproduces, are damaged. They don't work the way they're supposed to. And so these damaged cells can begin reproducing faster than the healthy cells around them. Normally, cells have mechanisms to tell when they're around another cell, and that tells them to stop growing. But these damaged cells have lost that ability, and so they grow and they pile up on each other, and that's where we get a tumor. Now, these damaged cells may produce too much or too little protein, or that protein may be deformed and just not work the way it's supposed to. And this over or underproduction of proteins, or these damaged proteins that might regulate how quickly a cell reproduces, leads to cancer or tumor growth. Now, I don't want you to think that cancer is a death sentence. It's not. We're coming up with new ways all the time to treat cancer. And there are cancers that are benign. They are not harmful. They can be taken out usually with surgery. But there are other cancers that are called malignant, and those are very dangerous because these tumor cells can then break away from this tumor and go to other parts of the body and seed new tumors. And that's when things become dangerous. So the quicker cancer is caught, the more likely a person is going to survive it. I hope that you learned something about why protein synthesis is important and how a protein synthesis works. These are my resources, especially the ones I used for pictures.